Hi, I'm Alden Cisnowski, and this year I'm taking IB Biology. For our project, we focused on the antibiotic cephalosporin, and I thought this was really interesting because I liked learning about how medicine really works and how it works on the microscopic level. During the project, I helped research, plan, film, and edit the video.
Hello, my name is Radira Vidal and I am in IB Physics. I was interested in cephalosporins because I wanted to learn about the diseases that affect people today and the antibiotics used to treat them. During this project, I researched the cephalosporins evolution and the mold it is derived from. I also helped plan and film the video.
Cephalosporin is an antibiotic, but what exactly does that mean? An antibiotic is a medicine that helps to cure infections such as pneumonia, skin infections, kidney infections, ear infections, strep throat, or any other disease caused by bacteria. Antibiotics are used every day by millions of people, but the most commonly used type of antibiotic is a beta-lactam antibiotic. This includes penicillin and cephalosporin. What makes these antibiotics so useful is that they target the cell wall of the bacteria, eventually destroying them. Cephalosporin, like penicillin, is a beta-lactam antibiotic. What this means is that in its chemical structure, there is always a beta-lactam ring, no matter the variation of the cephalosporin. There are many different variations of cephalosporins, each one performing a different function. All forms of cephalosporin share a similar base structure, including the beta-lactam ring, but different side chains give it different functions. However, each type all works in the same general way. It kills bacteria by breaking down the cell wall surrounding it. The way that cephalosporin accomplishes this is similar to penicillin, by limiting the production of peptoglycan, an important part in the cell wall of many bacterium. The lack of peptoglycan can cause the cell wall to weaken, eventually leading to the destruction of the bacteria. Because human cells don't have cell walls, an effective way to specifically target bacteria is by using something that targets its cell wall. This is exactly what cephalosporin does. Cephalosporin is a peptoglycan synthesis inhibitor, meaning that when the antibiotic is used, bacteria can't form new cell walls. This will eventually kill the bacteria when it attempts to perform cell division. As the cell division process starts, the cell wall is usually built up to contain everything. However, this is not possible due to the antibiotic limiting this growth. Eventually, the cell has doubled, but there is no cell wall surrounding the bacteria, leaving it vulnerable, and it will die very soon.
The evolution of cephalosporins. Cephalosporins were discovered in 1948 when Italian scientist Giuseppe Brazzo was looking at fungi from Sardinia sewers. He found cultures of Cephalosporium acremonium, which inhibits the growth of Salmonella typhi, which is what causes typhoid. In 1959, Guy Newton and Edward Abraham were able to isolate the nucleus of cephalosporins. Then, in 1964, cephalotine, the first cephalosporin, was released into the market and is still used today. Cephalosporins were created in generations, each generation showing a better ability to work against bacteria that test negative in the Gram test and are therefore harder to identify than positive results. The first generation was taken orally and treated simple skin and soft tissue infections, tonsillitis, and urinary tract infections. The picture is the chemical makeup. The second generation cephalosporins were used for microbial infections, such as ulcers and diabetic foot infections. Some were injectable. The picture is the chemical makeup of second generation cephalosporins. Third and fourth generation cephalosporins had many clinical uses, including their use to counteract polymicrobial infections, like second generation cephalosporins, as well as meningitis and pneumonia. Their chemical makeup was similar, if not exactly the same. The table shows the various subtypes of each generation. Finally, the fifth generation cephalosporins were specifically created in the lab to target resistant strains of bacteria. They have similar activity to that of third and fourth generation cephalosporins, but are used to treat skin and respiratory infections. There is the chemical makeup of fifth generation cephalosporins.
In our project, we learned about how a medicine can be discovered and how it can develop into one of the most commonly used drugs in the world. We also learned how cephalosporins work and why not all strands can combat each infection. Finally, we studied how scientists used chemistry to solve biological problems by altering cephalosporin through its 5 generations.